Okay, so in this video, we're going to review Unit 5 for the AP Bio test, which includes meiosis, Mendelian genetics, and non-Mendelian genetics. So when we look at meiosis, so in meiosis, it's a process that ensures the formation of haploid cells in sexually reproducing diploid organisms. So a haploid cell or a haploid gamete is a sex cell that has only one set of chromosomes. So in humans, for example, how we have 46 chromosomes, but we have 23 pairs. So a pair is made from like a chromosome inherited from the egg and a chromosome inherited from the sperm. So when we talk about this, the egg and the sperm each have one of every kind of chromosome. So the egg has 23 chromosomes and the sperm has 23 chromosomes in humans. Those would be our haploid gametes. And these are used in sexually reproducing organisms that are diploid. So when we talk about diploid means two sets of chromosomes. So when the sperm fertilizes the egg, you have two of each kind. If an organism is triploid, they would have three of each kind, like 3 number 1s, 3 number 2s, 3 number 3s. If an organism was octoploid, like a strawberry, they would have 8 number 1s, 8 number 2s, etc. So the other thing in the standard that says in sexually reproducing organisms, there are species that reproduce asexually, and asexually is basically reproducing by using mitosis or cloning. It does not involve the use of gametes or sex cells. So meiosis results in daughter cells with half the number of chromosomes of the parent cell, and this is to basically ensure restoring that diploid number. So when you have a like meiosis, the whole purpose of meiosis is to make gametes that are haploid. And so when you end with a haploid egg and a haploid sperm, when fertilization occurs, well, now you have a diploid organism. So it makes sense that meiosis would produce cells with half the number of chromosomes as a parent cell. So when a half plus a half equals a whole. And then meiosis involves 2 rounds of division, 2 rounds of a sequential series of steps. And when you think about the why there's 2 rounds of division, it's because in S phase of interphase, the DNA doubles. We make those sister chromatids. So it's kind of like, to me, it always trips me out a bit that we have a process that's creating haploid cells, but yet we double the DNA before it even starts. So in S phase, when we go through DNA replication and the chromosomes duplicate, in the first round of division in meiosis 1, we want to make sure that we separate all of those pairs of chromosomes. So how we talk about creating cells with half the number. So we start with diploid, and then we're going to divide all of those pairs apart in metaphase 1 and anaphase 1 in order to make sure that each gamete will get one of each kind of chromosome. But the problem is that the chromosomes are duplicated. So then in meiosis 2, that's very similar to mitosis, where now we're going to separate those sister chromatids. So we are having actual haploids with only one of each kind of chromosome. Okay, so when we look at mitosis versus meiosis, they are similar in the way the chromosomes segregate. And what that means is they use spindle fibers that attach to like the kinetochore or the centromere and pull the chromosomes apart. When we compare them, meiosis 2 is actually very similar to, wait, did I say mitosis? Meiosis 2 is very similar to mitosis. And Difference is that we have a different number of cells. So in mitosis, you get 2 identical daughter cells. They are identical not only to each other, but also the parent cell. And then in meiosis, we have 4 genetically different haploid cells. So not only are they different from each other due to crossing over and independent assortment, but they're also different from the parent cell. Meiosis starts with a diploid germ cell and ends with 4 haploid gametes. So the genetic content of the daughter cells, like the amount, is half or different from mitosis, but also they're not identical to each other thanks to crossing over and metaphase 1 and anaphase 1. Okay. So the separation of homologous chromosomes in meiosis 1 ensures that each gamete receives a haploid set of chromosomes that comprise both maternal and paternal chromosomes. So if we look, here is anaphase 1, but if we also look at metaphase 1 right next to it. So when we talk about, sorry, I hit pause on accident. When we talk about, like over here where the standard says comprise both maternal and paternal chromosomes. So this germ cell, this diploid cell we're starting with in interphase, is in a human. Like let's pretend it's in me, right? Well, I am the product of a gamete from my mom and a gamete from my dad. So technically my DNA is half maternal and half paternal. So if I were to look at this metaphase 1, we can pretend that these green chromosomes came from my mom and the purple chromosomes came from my dad. So you can see as my pairs line up along that metaphase plate, sometimes you'll have it's not always like maternal chromosomes on one side and paternal chromosomes on the other. This is random every time meiosis occurs, and there's actually like 8 million different possibilities of how our chromosomes align, like our homologous pairs align along that metaphase plate. So in prophase 1, our homologous pairs form. So like the chromosome number 1 I inherited in the egg from my mom will find chromosome 1 I inherited in the sperm from my dad, and they will pair up. And then this is where crossing over occurs. So I guess I should use like this. So here, like they're duplicated. So then they pair up in what we call a tetrad. So a tetrad is made of 4 sister chromatids, and this is where like crossing over occurs. And in crossing over, they'll exchange parts of their DNA. This like homologous pair forming does not occur in mitosis. This is unique to meiosis. And by having these pairs form, and then they move like to the metaphase plate. Now in anaphase 1, the pairs are actually going to separate. Now, every time meiosis happens, you get a different combination together. So like right here, we're only seeing these 2 that will end up together in a gamete. However, in a human, that would be 23 lined up along each side. And you get a different combination every time meiosis occurs. So let's pretend like this chromosome I inherited from my dad and this one I inherited from my mom. Then as I go through the process of meiosis and they separate, my offspring will only inherit one of these. They may inherit the one I got from my dad, or they may inherit the one I got from my mom. So my dad is Irish and German, and my mom is Scottish and Irish. So let's pretend that this is a like German descendant chromosome and this is a Scottish descendant chromosome. So my offspring is either going to inherit my Scottish DNA or my German DNA of this particular chromosome, but not both, because my offspring will be made from a haploid gamete from me, meaning that it has only one of each kind of my chromosomes. Now, which one it gets will be random each time, depending on independent assortment. So when we look back at the standard, the separation of homologous chromosomes in meiosis 1. my watch. So the separation of homologous chromosomes in meiosis 1 ensures that each gamete receives a haploid set of chromosomes that comprises both maternal and paternal chromosomes. So it'll be a combination. So my offspring will inherit some chromosomes I got from my dad and some chromosomes I got from my mom. So it'll be a mixture. And then it will also, though, from my partner, from the sperm, it will also, my offspring will also inherit like a mixture of those grandparents. So like my offspring is not only like the 4 grandparents DNA all mixed together, but like generations of ancestors all being like mixed together. Anyway, it's beautiful. Okay. So here when we talk about crossing, I liked this picture because it showed meiosis 1 versus meiosis 2 as far as the homologous chromosomes being separated. So in meiosis 1 is where like whole chromosome pairs are separated. Whereas in meiosis 2, this is similar to mitosis, where you're going to have your like identical sister chromatids separated. Except for now, they're no longer identical because of crossing over. So let me hit pause. I think someone's at my door. Okay, sorry. So during meiosis 1, homologous chromosome or chromatids, exchange genetic material via a process called crossing over or recombination, which increases genetic diversity among Rocco. Sorry, working from home is a bit tough. Okay, so what was I saying? So exchange genetic material via a process called crossing over or recombination, which increases genetic diversity among the resultant gametes. So recombination is basically like you can have recombinant chromosomes that are going to exchange pieces of DNA and then they recombine. So if you ever see like a recombinant chromosome, it means that it is the product of crossing over. Now, crossing over is also only going to, hopefully, only exchange like kind of like homologous segments of DNA as well. So like right here, if this was like the allele for eye color, so let's put like maybe it's brown eyes, a dominant allele, and over here is an allele for blue eyes. Like if it exchanges that set of DNA, it's the same set. Like the genes coding for eye color is switched with genes coding for eye color, not eye color switched with blood type or something. And so if you do have a mistake in crossing over, this could be a type of like a chromosomal mutation, like an inversion or something. Okay. So sexual reproduction in eukaryotes involving gamete formation, including crossing over, the random assortment of chromosomes during meiosis, and subsequent fertilization of gametes serves to increase variation. So this is kind of like a big deal in biology, is this idea of increasing variation. So variation is always a good thing in biology. Basically, like when we talk about offspring and offspring being genetically different from each other, that allows for natural selection and the survival of a species in changing environments. When you have identical clones for offspring, you lack the variation, which makes it difficult to survive in a rapidly changing environment or ecosystem. So variation is always good. And at least that I can think of. There may be some examples where it's bad, but I don't know any. So when we think about the 3 main sources of variation in sexual reproduction, so this is like separate from mutations, for example. Like mutations occur constantly and in both asexual and sexual reproducing populations. But when we talk specifically about sexual reproduction, you have crossing over creates unique and new combinations of DNA that have never existed. So, for example, you could take my stepdad. He is half Mexican and half Polish, right? So like he inherited Polish DNA and like Mexican DNA. So when that results in crossing over, like when he makes his sperm, he's going to have chromosomes that have like Mexican ancestry, plus like Polish ancestry, all on the same chromosome. Like it's going to be beautiful and a new and unique combination of genes that have never existed before could be forming his offspring. So anyway, crossing over creates beautiful new combinations of genes that have never existed. And yeah, I love crossing over. And then you have independent assortment, where with independent assortment, this is where when the homologous pairs line up along the middle of the cell, they could end up in different combinations, basically. And independent assortment is part of like one of Mendel's laws, I guess. I guess it is one of Mendel's laws, and it creates different combinations of gametes. And then we have the random fertilization of gametes. And so random fertilization, when you think about like all the possibilities that a human could make, like we have over 8 million different combinations possible with how our chromosomes will independently assort and then separate during meiosis. And so which of those gametes fertilize the egg and which end up in the egg is like unique every time. So you have random fertilization is also a third source of genetic variation in sexually reproducing populations. Okay. So the next couple slides I feel are more appropriate for Unit 7, which is our evolution unit, but that is okay. So when we talk about genetic carriers of information, so we have our like chromosomes getting passed on in our gametes, and so that would be our carriers of genetic information. And so when we look at this, so our chromosomes are going to carry genetic information from one like generation to the next. But we also have our plasmids in our mitochondria, or if we're like a plant, also has chloroplasts that have plasmids. So that genetic information is DNA, can be passed from mothers to their offspring. And then like how it says here, DNA and RNA carry genetic information. I was also thinking about like RNA viruses. As viruses replicate, like their genetic information is carried in the form of RNA. But we also have RNA carried, like carrying the protein sequences for like protein formation, protein synthesis too. Ribosomes are found in all life. So we find ribosomes that read our genetic information. Well, they don't really read it, but they read the mRNA to like build the proteins. Those are found in both prokaryotes and eukaryotes. And then our genetic information, our genetic code, is like evidence for common ancestry. It's found in all living systems. So from the smallest prokaryote to the largest eukaryote, our genetic code is universal. So AUG will always code for methionine. UUU will always code for phenylalanine, no matter the species. And this is what makes like genetic engineering possible, how we can take DNA from different sources and combine them together. We also have core metabolic pathways are conserved across all currently recognized domains. So from like prokaryotes to eukaryotes or archaeans, we have metabolic pathways like glycolysis or protein synthesis that are conserved and found in all species. So now we're going to go into Mendel's laws. I feel like I will stop the video here. So if people need a break or something, want to come back and watch the second part. So second part of Unit 5 review will be on Mendel. Well, I guess we'll just keep going. I'll edit that out. Okay, so here we're going to talk about Mendel's laws of segregation and independent assortment can be applied to genes that are on different chromosomes. So when you look at Mendel's law of segregation, it's basically saying that when we go through S phase and the DNA replicates, when we go through meiosis, these duplicated chromosomes are going to separate. So each gamete gets only one allele for each gene. They'll get one chromosome. So I am heterozygous for eye color. I have a dominant brown eye allele and a recessive blue eye allele, right? So but my gametes, half of my gametes will inherit that dominant allele and half of my gametes will inherit that recessive blue eye allele. So I have a 50-50 chance of passing the blue eye allele to my offspring or the brown eye allele to my offspring. And that is Mendels Law of segregation. The alleles segregate or separate during meiosis, so each gamete has only one. So when we make our Punnett squares, like that is what we're showing. We're showing the outcome of meiosis, going from a diploid organism to haploid organisms to set up our Punnett square. We are showing that at the top, these are the possibilities that could end up in the gametes. And now when we talk about Mendels Law of independent assortment, though, that's showing that there's different possibilities and different combinations. So here, if we look at like this dominant, if this is a dihybrid, so meaning heterozygous for two traits, you can see like dark purple and maybe this lighter color represents like white flowers. And then here you have like tall and short alleles. So a gamete could end up with a purple and tall alleles, but it could also happen where it ends up with a purple and a short. These are not linked genes. They are on separate chromosomes, and they're going to inherit like independently of each other. And so that's Mendels Law of independent assortment, that genes on different chromosomes inherit independently of each other. And we'll see this when we look at data and we compare it to linked genes. Okay, so we also have fertilization involves the fusion of two haploid gametes, restoring that diploid number and increasing genetic variation in populations by creating new combinations of alleles in the zygote. So we can use rules of probability to analyze the passage of single gene traits from parents to offspring. So when we talk about probability, it's really like, what is the likelihood of something happening? So here, like you can use the rule of multiplication, like the chance of this happening and this, you would multiply. But if you are using like the rule of addition, then you would be adding together. So for example, in this yellow Punnett square, like what is the chance of a child inheriting the dominant phenotype? It would be like 1/4 plus 1/4 plus 1/4 would be 3/4. The rule of multiplication really comes in handy when you're talking about multiple genes and multiple traits. And so I have YouTube videos that show like solving dihybrid or two trait crosses using the rule of multiplication. I think that is a super helpful thing to know how to do. Okay, and make your life a lot easier when you are doing complicated problems in a rushed amount of time. So the pattern of inheritance. So let's talk for a minute right now about the pattern of inheritance. So basically, you can inherit traits on autosomal chromosomes, which are your chromosomes 1 through 22, where you are diploid. You have two number 1s, two number 2s, two number 3s, two number 4s, etc. And you have sex chromosomes. Your sex chromosomes are like your X's and your Y's. And so as a female, you have two X's, but a male has an X and a Y. So those traits show a different pattern when you look at data. So the pattern of inheritance, you can inherit a trait as autosomal dominant. For example, that would be something like dimples or brown eyes. You can inherit a trait that's autosomal recessive, which would be like maybe Tay-Sachs, which is like an inherited recessive disease. It's on an autosomal chromosome and it's recessive. You could inherit an X-linked dominant trait or an X-linked recessive trait. Like those are your 4 options for patterns of inheritance. And you can predict these by looking at data. So if you see a 3-to-1 ratio of a trait, you know, like in this data here, like all like round versus wrinkled, when you look at like the F2 ratios and you get a 3-to-1, when you cross like a round with a round and you get a 3-to-1 ratio round to wrinkled, that's telling you that round is dominant and those F1 are all heterozygotes. Versus if you do like at a 1-to-1 ratio, for example, that would tell you that one parent's heterozygous and one parent's homozygous recessive. So you want to be familiar with reading ratios and then making inferences from that to know what the parents' genotypes were. Okay, sorry, I had to hit pause. I'm not really sure where I left off. My family came home. So let's go ahead. So anyway, I think I was saying something about pay attention to ratios and put some effort, because that'll make your life easier when you're looking at data and trying to find patterns to figure out how traits are inherited. Okay? Okay. So here, genes that are adjacent and close to one another on the same chromosome may appear to be genetically linked. The probability that genetically linked genes will segregate as a unit can be used to calculate the map distance between them. And so this is a bad like Google image that, well, actually I think these images are from Khan Academy. I really like Khan Academy's pictures. But when you look at like finding map distances, so first off, when we talk about genes being inherited on separate chromosomes, like that is Mendels Law of segregation, and then Mendels Law of independent assortment, that these traits do not influence each other when inherited. But like ideally, like if you were looking at genes on different chromosomes and trying to calculate the probability of them being inherited, like what's the chance of inheriting a dominant A and a dominant B, or a dominant A and a recessive B? If the traits were on different chromosomes, they would have like equal likelihood, as you can see here, like a 25% chance, 25% chance, 25% chance, 25% chance, which is what we write at the top of like a dihybrid cross. However, sometimes if the traits are on the same chromosome, they would not be like segregating like independently from each other. They would be going together. But here we see on this right-hand side, sometimes if genes are really far apart from each other, crossing over happens so often that they actually will, like if crossing over happens more than 50% of the time, then we can like talk about those traits as if they actually were on separate chromosomes. So if you find a crossing over frequency of greater 50% or greater, you can treat those traits as if they were not linked. So linked though would be something like this. If you had like that recessive, like the A allele here, and then right really close by, you have the B allele. The chance of crossing over, like separating those, that is calculated based on like recombinant offspring. So here, if these traits on these flies were on separate chromosomes, the likelihood of each of these phenotypes would be equal, 25%, 25%, 25%, 25%. But you see the data is not equal. You see these offspring here that have very small amount are non-parental offspring, like they don't look like either parent. Those are your recombinant offspring. Those are the results of crossing over. So you can actually calculate like recombination frequency, like how often crossing over happens, by adding up your recombinant offspring. So 151, oh my gosh. Oh man, you basically add up, I'm not really good at writing on it, but 151 plus 154 divided by your total offspring, and that'll tell you like how often crossing over happens. So based on how often crossing over happens, like if we took a look at A to B and B to C right here, this 13.2 is telling you that 13% of the time crossing over occurs between A and B. And then this 6.4 is saying that like 6.4% of the time crossing over separates B and C. So the fact that like 6.4 is small, smaller than 13. That tells you that B and C are closer together, and then A and B are further apart. I think I have other videos on this that hopefully do a better job explaining it. Okay. So some traits are determined by genes that you find on sex chromosomes and are known as sex-linked traits. So when we talk about sex-linked, we're pretty much talking about genes on the X chromosome. The Y chromosome in humans has only about 30 genes on them, whereas the X has about 1,000 genes. And so generally sex-linked means X-linked, but you could also have, like in some reptiles and birds, they have a ZW system of like sex-linked. So instead of X's and Y's, they use Z's and W's. And so just because here we talk about X's and Y's, this can apply to other sex chromosomes such as Z and W, but they are chromosomes and they do carry genes. And the difference in sex-linked traits is that here, like in humans, the male inherits only one X, and they get that X from their mother and the Y from their father. So if you have a male offspring with a trait that's X-linked or sex-linked, he inherited that from his mother. And like in this pedigree here, the fact that both of these daughters have the trait is one sign that it's a sex-linked dominant trait, because the father, oops, oh my gosh, Mary. The father's only has one X to give his daughters. And so like here, this female would be X-linked, like female 6 right here, would be X-linked recessive. And so she would be giving recessive X's to her daughters. So both her daughters would be heterozygous. And you can see here how both sons have the recessive allele. Okay. Oh my gosh. All right. So I would recommend practicing with pedigrees. Now, some traits are the product of multiple genes and or physiological processes acting in combination. These traits, therefore, do not segregate in Mendelian patterns. So sometimes our traits aren't necessarily as cut and dry as dominant and recessive on an autosomal chromosome. So a good example is our skin color. Skin color is actually polygenic, meaning it's the product, like our one phenotype is the product of multiple proteins working together inside of us. So here you can see if you have two people who are heterozygous for all 3 genes dealing with skin color, this Punnett square is showing the probabilities or outcomes of like all the different possibilities of skin color in their children. So it is, when we talk about like the product of multiple genes, skin color is the example of that. But then if you also think about and or physiological processes, like if you look at like height in humans, height has a genetic component, but so does nutrition. Nutrition plays a role in height. If a person is malnourished, they're not going to grow to their full genetic potential. So sometimes our phenotypes are not only due to genetics and proteins. Now, some traits are from non-nuclear inheritance. So non-nuclear inheritance is referencing DNA outside of the nucleus, which is basically our plasmids that you find in mitochondria. And then in plants, you would find plasmids in both mitochondria and chloroplasts. So these do not follow meiosis with haploid gametes and stuff. And instead, when we think about how mitochondrial DNA is inherited, it's inherited from the mother to her offspring. It's the egg that is the source of all organelles in offspring, so therefore the mitochondria goes from mother to children. So I truthfully have 2 sons. So both of my sons have my mitochondrial DNA, but my grandchildren will not, because my sons will only contribute their haploid DNA to my grandchildren. So my mitochondrial DNA stops with me. All right. And sometimes environmental factors can influence gene expression and can lead to phenotypic plasticity, which means like a flexibility in our phenotypes. Phenotypic plasticity occurs when individuals with the same genotype exhibit different phenotypes in different environments. So here you can see like stomata grown in different carbon dioxide or plant leaves grown in different carbon dioxide concentrations will have different amounts of stomata. Or if you look at these grasshoppers, also depending if they were grown or raised by themselves or in a crowded population, it actually changes their gene expression. So gene expression, like when we think about, wait, hold on, Rocco, can you please stop? When you look at your your phenotype, when we keep talking about alleles that you inherit, your DNA sequence codes for a gene. And then your gene is transcribed and translated and forms a protein. So it's your proteins that ultimately determine your phenotype. My brown eyes is because of a particular protein in my eyeball. My skin color is due to the protein melanin, right? So like my hair is keratin, which is a protein. Like our phenotypes are based on proteins that are coded in our genes. So how we express those genes is gene expression, and that is also dependent on environmental factors. So this ties a lot into Unit 6 that deals with transcription factors and turning on gene expression. Okay. Segregation, independent assortment and of chromosomes and fertilization result in variation. Like we mentioned earlier, variation is really important. And then here, the chromosomal basis of inheritance, meaning that understanding that it's chromosomes that are passed from one generation to the next, that gives us an understanding of the pattern of how genes go from parents to offspring. And then our last slide here talks about sometimes there can be errors in meiosis and when we form these gametes. So certain human genetic disorders can be attributed to the inheritance of a single affected or mutated allele, or so like we talk about human genetic disorders like cystic fibrosis, Tay-Sachs, PKU. Like these are genetic diseases or even like hemophilia that are due to like a single allele that is affected. So sickle cell anemia, cystic fibrosis, Tay-Sachs, these are due to mutations of a certain gene at a particular locus. A locus is like a location. Or sometimes human genetic disorders can be to abnormal number of chromosomes, which are the result of non-disjunction. So in non-disjunction, this is where, like in meiosis 1, homologous pairs fail to separate. So here you have some chromosomes, some gametes that have like extra chromosomes or missing chromosomes. And so meiosis 2 can also have non-disjunction where your sister chromatids fail to separate, also resulting in an abnormal number of chromosomes. So like, for example, my grandma's brother had Down syndrome, and that would be the cause one of his gametes, either from my grandma's mom or my great grandmother or my great grandfather, produced a gamete that had an extra chromosome 21. So therefore my great uncle, Uncle Buddy, he had an extra like 3 copies of chromosome 21. And that would be the result of non-disjunction, either in meiosis 1 or meiosis 2. And the chances of non-disjunction do increase with the mom's age. So as a female ages, the older she is having offspring, the higher the likelihood of non-disjunction. But Down syndrome is not the only example of non-disjunction or having an extra set of chromosomes, or an extra set, not extra set, sorry, an extra. If it was an extra set, then you would have like 3 number 1s, 3 number 2s, 3 number 3s, 3 number 4s. That is not what we're talking about. We're talking about just one extra, like an chromosome number 21 has 3 copies, but like chromosome number 1 has 2 copies. Chromosome number 2 has 2 copies. Chromosome number 3 has 2 copies. It's just only like 1 extra chromosome, not a whole 23 extra. Okay, I want to be clear on that. A few of my students mixed that up when they were writing their FRQ. Anyway, okay, so this is Unit 5, and sorry it's kind of a long video. I apologize for some of my children and dog and a little bit of disruptions there, and I hope that it was helpful for you, whether you're using it to prepare for the AP test or a unit test. I wish you the best of luck. Good job.